Howdy, my name is Isabel Navarro, and I'm excited to share my presentation today on how seasonal bloom timing influences forage quality and availability for Apis mellifera colonies, which is a major topic in honeybee biology. Apis melliferus, or the western honeybee, is one of the most important pollinators for both agriculture and ecosystems. For honeybees, their colony health is deeply connected to the availability of quality forage like nectar and pollen from blooming plants, and as climate changes and bloom timing shift, it can create a mismatch between the bees' nutritional needs and available forage, leading to stress in the colonies. Why does bloom timing matter to Apis mellifera? Well, in this section, we will dive into why bloom timing is so crucial for honeybees colonies, Apis mellifera, which plays a key role as a pollinator for many of our crops and wild ecosystem. Without them, we would face massive reductions in biodiversity and food security. For honeybee colonies to thrive, they depend heavily on the availability of quality forage. Forage is primarily made up of nectar and pollen from seasonal blooming plants, and the timing of those blooms directly affect the health and productivity of the colony. Any changes in bloom timing, often driven by environmental shifts or climate change, can cause a misalignment between the bees' nutritional needs and what's available to them, leading to colony stress. Now, let's look at the background of the bloom phenology and how climate impacts bloom timing. Bloom phenology is essential to the study of timing of plant flowering in response to environmental cues like temperature and rainfall. We've seen that climate change has shifted bloom timings for many plants, disrupting the availability of forage for honeybees. This shift can create a forage gap when bees have to deal with periods of low nutrition, and when these gaps are long enough, the colony suffers. For Apis mellifera, a continuous supply of diverse forage is essential, especially during the spring and summer months. Our research question here: how does seasonal bloom timing influence forage quality and availability for Apis mellifera colonies, and what are the consequences for colonies' health and productivity? With all this background in mind, our research question focuses on understanding how seasonal bloom timing influences forage quality and the availability for Apis mellifera. Specifically, how does the timing of bloom impact colony health, and what are the long-term effects on colony productivity? We also explore how the shift in bloom timing caused by climate change leads to challenges for the honeybee colonies, making it harder for them to maintain their health and productivity levels. In this first experiment, we looked at the diversity of pollen types collected by bees across floral diversity in mid-spring. With a steep decline in late summer, this has serious implications for the colony's health because reduced floral diversity leads to nutritional bottlenecks, affecting the colony's ability to rear brood. You can see from the graph that in the early months, the bees collected a greater variety of pollen, which is critical for maintaining healthy and well-balanced nutrition. But as we move into the summer months, the pollen diversity significantly decreases, which leads to nutritional stress on the colony. Moving on to experiment 2, we focused on analyzing the protein and lipid contents in pollen collected across season. This study found that spring pollen tends to be richer in essential amino acids and fatty acids, which are vital for honeybee nutrition and immune support. However, as we approach late summer, the nutritional content of pollen declines, leading to a drop in protein levels, which directly impacts the bee's immune function and overall health. The graph here illustrates the comparative nutritional values of pollen collected during different seasons. This shows us that the decrease in pollen quality, especially in the later part of the year, has detrimental effects on the colony. For experiment 3, we observed how bloom gaps, which occur when there is a lack of flowering plant during certain parts of the season, affect colony weight and brood production. The study tracked hive weight across the bloom season, and we found that when there are significant bloom gaps, there is a noticeable decline in hive weight, which signals a decrease in the colony's overall health. As you can see from the line graph, the hive weight drops during periods of bloom gaps, indicating a lack of nutrition for the colony. Supplemental feeding is often needed to support these bees during these times, but if the gaps are prolonged, it can lead to long-term stress on the colony. The implication of the research are significant. Oh, I'm so sorry. To summarize, seasonal bloom timing plays a direct role in determining the availability and quality of forage for Apis mellifera's. Spring provides the optimal nutrition for the colony with a wide variety of pollen types, but late season shortage can cause nutritional stress, impacting brood production and colony health. Floral diversity and bloom synchronization are absolute critical to maintaining healthy and resilient colonies. The implication of this research was significant. The findings emphasized the importance of habitat restoration efforts, specifically planting diverse and sequentially blooming flora to provide a continuous food source for honeybees. The result also highlighted the need for phenological monitoring in apiary management, as well as the conservation of native flowering plants to support healthy honeybee population. These actions are essential to mitigating the effects of climate change and ensuring long-term success for honeybee colonies. Lastly, I would like to acknowledge the sources that have contributed to this research. Here's a list of the reference used in this presentation, which includes peer-reviewed journal articles and studies providing the scientific foundation for our discussion today. Thank you for your attention, and I hope this presentation provided you with a valuable insight into how bloom timing impacts honeybee health and what we can do to support these critical pollinators.